Chapter 3 is the cellular system, and we're going to learn about how the cells are important in our body. So we know that cells are the smallest unit of life and that they are classified as a living thing. So we can go back and we know that there's two types of cells. There's prokaryotic and there's eukaryotic. And we learn things about cells in biology, but specifically this chapter is how our cells how are they related to our body. In other words, we need to understand the transport mechanism through our cells, what protects our cells, all of the structures and the cell components. That's what we're going to get into during this chapter. So cells are the smallest unit that can carry on all life processes, and they are the building blocks of all living things. So they're very, very important, and we have trillions, approximately 75 trillion cells just in our human body. So the cell structure consists of three main regions: the plasma membrane, the nucleus, and the cytoplasm. So we're going to dive into each of these separately. So let's start with the plasma membrane. It functions as a barrier for cell contents, separating what's outside from what's inside of the cell. So it controls all the cellular activities through the proteins embedded within it. So again, it is surrounding the cytoplasm and the nucleus, and it is physically separating the intracellular components from the extracellular environment. So the proteins embedded carry out specific functions, including selective transport of molecules and even cell-to-cell recognition. So the cell's outer membrane is made up of two layers of phospholipids, and again with embedded proteins within it. So it separates the contents of the cell from what is in the outside environment. It regulates what enters and even exits the cell. So the plasma membrane is very, very important for our cell. The structure of the plasma membrane is composed of a double layer of phospholipids, also called a lipid bilayer. So that means two layers. So again, it's the outermost covering of the cell, and it is very protective of what's inside of our cell. And that's good because that's going to help us maintain homeostasis of our cell, because it is this double layer of phospholipids, and it is protecting what is entering or leaving the cell. So a term that you may be familiar with is selectively permeable. This selectively permeable means that the plasma membrane allows some materials to pass while excluding others. Now, permeability is dependent on size and solubility of the material passing through it. So here the phospholipids make up the double layer, which is the phospholipid bilayer. So, and it is semi-permeable. So they have a phosphate head and two fatty acid tails. Now, the hydrophilic phosphate head is on the outside layer and is exposed to the water content outside and within the cell. The hydrophobic tails are the layer hidden inside of the membrane, and the phospholipid bilayer is most permeable to small uncharged molecules. So being selectively permeable, this is essential to maintaining homeostasis because it's regulating the passage of some substances while preventing others from entering the cell. Now, the plasma membrane has several things involved in its makeup, I guess you could say, in its framework. And what some of these are cholesterol, and this has a stabilizing effect and keeps the membrane secure. Now, cholesterol, it maintains the integrity of the membrane. So it plays a role in facilitating cell signaling, such as cell communication, and it makes up nearly half of the cell membrane. Due to its shape, it does help slightly immobilize the outer surface of the membrane and make it less soluble to very small water-soluble molecules that could otherwise pass through more easily. Enzymes are involved in chemical reactions. So we're going to talk a little bit more about enzymes in each chapter. Well, we mentioned enzymes in chapter 2, the chemistry chapter. So we learned how enzymes are used to start chemical reactions without being changed themselves, and they can go on and start other reactions in the body. Receptors are communication sites, specific binding sites for hormones and other chemical messengers. So in order to respond to changes in their immediate environment, cells must be able to receive and process signals that originate outside of their borders. So how do cells recognize these certain signals? Well, the cells have proteins called receptors that bind to signaling molecules and initiate a physiological response. Now, different receptors are specific for different molecules, so just such as insulin receptors binds insulin, for example. And so receptors bind to molecules that can pass through the plasma membrane. Next, we have transport proteins. These are channels that allow substances to enter and leave. So this allows for selective passage of specific molecules from the external environment, and each protein is specific to a certain molecule. Then we have glycoproteins. These are sugar proteins. They function as receptors. They determine our blood type. So example in the body is such as your mucins. This secreted in the mucus of the respiratory and digestive tract, for example. So that is just some of the contents here that you'll find embedded within our plasma membrane, and they all have different roles. And again, all of those roles help to maintain homeostasis of each of our cells. Some specializations that you'll see with the plasma membrane, we have something called microvilli. This is finger-like projections that increase surface area for absorption. So they are involved in absorption, secretion, cellular adhesion, and they form the brush border that is found on the apical surface of some of our epithelial cells, such as your small intestines. Then we have some membrane junctions that are specialized proteins that connect to other cells. So you have your tight junctions that hold adjacent cells tightly together so nothing can penetrate between them. You have your desmosomes and your tight junctions. Kind of keep in mind that they act as a barrier that regulate the movement of water and solutes between epithelial layers. Your desmosomes hold these adjacent cells together by fibers, but they can be separated. Then you've got gap junctions. These are open areas in the membrane allowing the passage of chemicals between cells. So your gap junctions are kind of like your communicating junction. They allow for direct chemical communication between adjacent cellular cytoplasm through diffusion without the contact of extracellular fluid. So those are some of our membrane junctions. Next, we get into the nucleus. The nucleus controls cellular functions. Now, the function of the nucleus, of course, is to also maintain the integrity of the genes and control activities of the cell. So our DNA, we find our DNA here. This is our genetic material containing information for making proteins. Chromatin is a form of DNA, and 90% of the time it consists of the DNA and proteins. It's very stringy and fibrous. Then you have your chromosomes. Condensed chromatin forms during cell division. So our nucleus was the first organelle to be discovered, actually. And something else about the chromatin here is that you need to understand it's a combination of DNA and proteins that make up the contents of the nucleus. 3 functions of chromatin is to, 1, package the DNA into smaller volumes to fit into the cell. And then secondly, to strengthen the DNA. And then third, to prevent DNA damage. And then our chromosomes, kind of keep in mind that that's just a single piece of coiled DNA containing many genes. So you have also the nuclear envelope, and keep in mind your nucleus is kind of known as our cell's control center. So your nuclear envelope is a barrier of the nucleus separating it from the rest of the cell. So that's very similar to the cell's actual plasma membrane because it is a barrier separating it from the rest of the cell. You have the fact that it surrounds the nucleus, and it also has this double membrane with multiple pores. You have the nuclear pore, that is openings within the envelope that allow for the exchange of material with the rest of the cell. Now, the nuclear pores, they regulate the passage of macromolecules like proteins and RNA, but they're going to permit free passage of water, ions, ATP, which is our energy, and other small molecules. Then you have the nucleolus that's composed of your RNA and proteins and function as the site of ribosome production. Your nucleolus is a non-membrane bound structure found in the nucleus of our cells, and its function is to, again, transcribe ribosomal RNA and combine it with proteins to form almost complete ribosomes. Now, it occupies about 25% of the volume of the nucleus, and a malfunction of it can be the cause of several human diseases. Next, we move into our cytoplasm. Our cytoplasm is the material outside of the nucleus, but yet inside of the plasma membrane. We have the cytosol. Well, let me go back first of all on the cytoplasm. Let me mention to you, it is kind of clear in color. It has a gel-like appearance, and it is composed mostly of water, and it actually helps to move materials around the cell and also dissolves cellular waste. The cytosol is the fluid that suspends other elements. So this is the portion of the cytoplasm not contained within membrane-bound organelles. It makes up about 70% of the cell volume. It is part of the cytoplasm that is not held by any organelles. And then our organelles are tiny organs within the cytosol that perform specific functions. So we're going to get into each different organelle and discuss their individual function. The first organelle that we're going to talk about is the ribosome. The ribosome is the site of protein synthesis. It is the location within the cell. You have two different locations. You can have ribosomes free floating in the cytoplasm or attached to the rough ER. Now, it does consist of RNA and proteins. It is responsible for assembling the proteins of the cell, and the cells need to make proteins because they might be used as enzymes or as support for other cell functions. Protein synthesis is the formation of proteins. So although DNA stores the information for protein synthesis and RNA carries out the instructions encoded in the DNA, most biological activities are carried out by our proteins. So there are three types of RNA, and all three play a role in protein synthesis. We have the messenger RNA that carries the information from the DNA. Again, we get the DNA from the nucleus to the ribosomes. Then we have ribosomal RNA. That's a type of RNA that forms the ribosome. And then we have transfer RNA that carries amino acids to the ribosomes. So with protein synthesis, each of these RNA types play a different role. And the messenger RNA specifically, again, it just carries a copy of instructions for the assembly of amino acids into proteins from the DNA to the rest of the cell. Now, in a series of 3 base code words, each of which specifies a particular amino acid. We're not going to get into making an amino acid. I just want you to know how it is formed. The ribosomal RNA, again, associates with a set of proteins to form ribosomes, and they catalyze the assembly of amino acids into the protein chains. And then your transfer RNA, this is the key to deciphering the code words in the messenger RNA. So each type of amino acid has its own type of transfer RNA, which binds it and carries it to the growing polypeptide chain if the next code word on the messenger RNA, of course, calls for it. Now, transcription and translation. Transcription is information in the DNA is copied onto the messenger RNA, and it does occur in the nucleus. And then, so with transcription, this process involves transcribing genetic information from DNA to RNA. The transcribed DNA message is used to produce proteins. Now, DNA is housed within the nucleus of our cells. It controls cellular activity by coding for the production of proteins. So it is the first step of gene expression, and it does occur in the cell's nucleus where the DNA is at. Then you have translation. The information in the messenger RNA is used to link amino acids together, forming a protein. So translation is the whole process by which the base sequence of a messenger RNA is used to order and to join the amino acids in a protein. So those three types of RNA, they all three participate in this essential protein synthesizing pathway in all of our cells. So translation, essentially we have protein synthesis being accomplished through this process occurring in the cytoplasm. And this occurs when the messenger RNA strand moves out of the nucleus and into the cytoplasm. And again, the process in which cellular ribosomes are creating proteins. We have the endoplasmic reticulum. So the endoplasmic reticulum is basically a network of tubules and flattened sacs that serve a variety of functions in the cell. Now, many membranes found throughout the cytoplasm, and again, they do function as channels for the transport of materials. You have two types. You have the rough ER and you have the smooth ER. The rough ER contains ribosomes and is associated with making proteins because of the ribosomes. So the rough ER manufactures membranes and secretory proteins. In the white blood cells, the rough ER can produce our antibodies. In the pancreas, our rough ER produces insulin. So it is very important in synthesis and packaging of our proteins. The smooth ER lacks ribosomes and it functions in the synthesis of lipids such as cholesterol, fat metabolism, and capable of detoxifying. So this smooth ER serves as a transitional area for vesicles that transport the endoplasmic reticulum products to various destinations in the liver, and it helps to detoxify in the muscles that assist in contraction even of our muscle cells. The Golgi apparatus functions as a factory in which proteins received from the endoplasmic reticulum are further processed and sorted for transport to their destinations as lysosomes, plasma membrane, or secretion. So they process, package, and secrete proteins. So kind of think of this as your cell's post office system. It sorts, processes, and packages materials to be transported within the cell or to be excreted. So we process, the Golgi finishes assembling the protein, we package, a new vesicle is formed, and then we secrete. The new vesicle is sent to another destination within the cell or to the plasma membrane to be released. So again, this is the organelle that builds our lysosomes. So those lysosomes are our cell digesting machines. So that's kind of what our Golgi does for our body. And then we have the mitochondria. The mitochondria is known as the powerhouse of the cell where cellular respiration takes place. Now we have the matrix, that's the thick fluid inside the mitochondria, and we have the cristae, which is the inner membrane foldings containing enzymes that function in cellular respiration. So the mitochondria convert energy into forms that are usable by the cell. Also, they're involved in cell division and growth as well as cell death. So these are our organelles that act like a digestive system that takes in nutrients, breaks them down, and creates energy for the cell. So this process of creating energy, this is known as cellular respiration. Now, our mitochondria, we need to understand that cellular respiration requires oxygen to break down organic nutrients to release energy. So we get that energy from our mitochondria. That's why it's called the powerhouse of the cell. Cellular respiration is the process of breaking down glucose and oxygen into carbon and water while releasing energy in the form of ATP to do cellular work. The process of cellular respiration, we have glycolysis and we have the Krebs cycle. Now, glycolysis breaks down glucose into pyruvic acid and releases 2 ATPs. It does not require oxygen, so it is known as anaerobic respiration, and it does take place in the cytoplasm. So it literally means splitting sugars. So it allows cells to make small amounts of ATP, and this process can be called fermentation. The Krebs cycle is also known as the citric acid cycle, and it breaks down pyruvic into carbon dioxide and water while releasing 34 ATPs. It requires oxygen, so it's called aerobic respiration. So in eukaryotic cells, this takes place in the mitochondria. It generates a pool of chemical energy from the oxidation of pyruvate, which is the end product of glycolysis. Our lysosome Our lysosomes contain digestive enzymes and are used to clean up the cell, digest bacteria that have entered the cell, or break down worn out organelles. So it is a membrane enclosed sac that contains digestive enzymes. Your lysosomes, they hold our enzymes that were created by the cell. Its purpose is to digest things. It might be used to digest food or break down the cell when it dies. Enzymes were first created in the rough ER, and those proteins are packaged in a vesicle and sent to the Golgi apparatus. The Golgi then does its final work to create the digestive enzymes and pinches off a small vesicle, and this vesicle is a lysosome. So some digestive functions of our lysosome: they serve as food vacuoles, the cytoplasmic sac that contains nutrients. Lysosomes digest the nutrients for our cell nourishment. Worn out organelles and proteins, something called apoptosis. This is genetically programmed cell death, so it destroys abnormal cells, destroys cells during embryo development. So when the cell is already abnormal and going through abnormal growth formation, this programmed cell death usually occurs automatically because our body recognizes it as a foreign invader. And then also the bacteria with your white blood cells also is a way that we can serve as these digestive functions. Peroxisomes are membranous sacs of oxidase enzymes. They break down free radicals. And what are free radicals? These are atoms within a cell that have unpaired electrons in their valence shell, and they can change a protein. Peroxisomes are small membrane enclosed organelles that contain enzymes involved in a variety of metabolic reactions. Peroxisomes are organelles found in all of our eukaryotic cells. The cytoskeleton, this is, just think of this as your structure, just as your skeletal system of your body. This is the skeletal system of our cell. So we have microtubules and microfilament fibers extending throughout the cytoplasm and functioning in the shape and support of the cell along with cell movement. So this is a network of fibers throughout the cell's cytoplasm that helps the cell maintain its shape and gives support to the cell. The centrioles are rod-shaped bodies made of microtubules, and they are the direct formation of the mitotic spindle during cell division. So the spindle will separate in the chromosomes during cell divisions. So the centrioles are basically there in the cell to help the cell when it comes time to divide. So they go to work during mitosis and meiosis. So we're going to find them near the nucleus, and they cannot be seen when the cell is not dividing. So when the cell is not dividing, you will see a condensed and darker area of the cytoplasm. That's called the centrosome. So when it comes time for cell division, the centrioles will appear and move to opposite ends of the nucleus, and the centrioles will duplicate. The cilia and flagella, these are motile appendages, and flagella has this whip-like tail that propels the cell. And an example of that would be our sperm cell. And the cilia that are motile are found in the lungs, the respiratory tract, and even your middle ear. So they work to keep the airways clear of mucus, dirt, allowing us to breathe easily without irritation. Again, they help propel sperm cell. And non-motile cilia act as a sensory antenna for the cell, so receiving signals from other cells or fluids nearby. So in the kidney, for example, cilia will bend with urine flow and send a signal to alert the cells that there is a flow of urine. So the flagella, such as your sperm cell, and the cilia, they're a little different. Like I said, the flagella has that whip-like tail that propels the cell like a sperm cell. Your cilia are those hair-like projections that move in a coordinated back and forth motion, such as, like I said, I talked to you about the lungs, respiratory tract, the middle ear, the kidney, and again, the flagella, we have more of a motion, like a locomotion here. And again, the main function is to help the cell move. So now let's get into transport through the plasma membrane. So we've talked about the plasma membrane of our cell. We've talked about what components are in the cell. So let's talk about how we can get across the cell membrane, or the plasma membrane, into the cell. So we have passive transport requires no energy, and those are diffusion, osmosis, facilitated diffusion, and filtration. And then we have active transport, which does require energy. ATP is our source of energy, and that is our sodium potassium pump, endocytosis, and exocytosis. So let's start with passive transport of diffusion. So this is the process by which molecules spread from an area of greater concentration to an area of lesser concentration. So we have a concentration gradient that must be reached, and that is where we find a difference in the amount of molecules in different areas. So then we strive to find equilibrium. So that is when the concentration of the molecules of a substance is the same throughout a space. So the particles evenly disperse until equilibrium is reached. Now, diffusion is going to continue until the concentration of substances is uniform throughout. So an example might be gas exchange at the alveoli, oxygen from air to the blood, carbon dioxide from blood to the air, and gas exchange for photosynthesis, carbon dioxide from air to a leaf, oxygen from leaf to air. So different things here. This picture that we have here on this slide is just like a food coloring tablet placed into a beaker of water, and there is a concentration gradient, but then equilibrium is reached. Another passive transport is osmosis. So osmosis is the diffusion of water. The direction of water movement depends on the concentrations of water and solutes dissolved in the solutions. So an example of osmosis would be like absorption of water by plant roots, or absorption of water by the stomach or the small intestines or the colon. So osmosis is just, again, simply diffusion, but it is just the direction of water. Now we have hypotonic solution, and we're still talking about passive transport osmosis here, but we have hypotonic solution. This is where the environment has a lower concentration of solute, so the water is going to move into the cell. Then we have hypertonic solution. The environment has a higher concentration of solute, so the water is going to move out of the cell. And then we have an isotonic solution where there is an equal concentration of solute in the environment, so water is going to move in and out at equal rates. So this picture here depicts hypotonic, hypertonic, and isotonic. So when we look at these, the hypotonic, again, we are flooding the cell, causing it to swell, and ultimately the cell can only take so much water and it's going to burst. So you need to understand the term lyse, L-Y-S-E, and that means to swell or burst. So again, it's kind of like a water balloon. You can only fill it with so much water until it ruptures. Then you've got the hypertonic solution, where we are losing water from the cell. So you can see the arrows going out. So again, we're losing water from the cell. And that term is called to crenate, C-R-E-N-A-T-E, to crenate. And that just means to shrivel and shrink due to loss of water. And then you have isotonic solution, where there is no gain or loss of water by the cell. So that's equally, the water's moving equally rates in and out of the cell. And that's, of course, ideal. That is the environment that we would want for all of our cells. Is an isotonic solution, because homeostasis and equilibrium is being met. Passive transport facilitated diffusion. This is just simply the process of diffusion that requires a transport protein. So for facilitated diffusion to occur, it does require a gradient and a transport mechanism, and such as like movement of glucose across the plasma membrane is achieved by facilitated diffusion. Passive transport filtration is another example of passive transport requiring no energy at all. Water and solutes are forced through a membrane by hydrostatic pressure in the body. Filtration is powered by our blood pressure. So This is important in the kidneys, where water and solutes filter from the blood into the kidneys because the pressure in the capillaries is greater than the fluid in the kidneys. Then we switch gears and we get into active transport. Now remember, active transport does require energy. You must have energy. So that's ATP. So ATP is used in this transport process. We have carrier proteins. The molecule entering or exiting first binds to a receptor on a carrier protein, and ATP is used to change the shape of the carrier protein, allowing the molecule to be released on the other side of the membrane. Then you have the sodium-potassium pump. This allows sodium to exit the cell and potassium to enter the cell. So it is necessary for muscle contraction and impulse conduction. So the sodium-potassium pump, the process of moving sodium and potassium ions across the cell membrane is an active transport process involving the hydrolysis of ATP, which is our energy, to provide that energy for the cell. Now, this is responsible for maintaining the large excess of sodium outside the cell and the large excess of potassium ions on the inside. So again, the sodium-potassium pump simply allows sodium to exit the cell and potassium to enter the cell, and it is necessary for muscle contraction and impulse conduction. Endocytosis and exocytosis. Here we talk about, again, we're still on active transport, talk about materials being too large to enter or exit through carriers. So they move through the plasma membrane using vesicles. So exocytosis moves substances out, and endocytosis moves substances in. So you can see there how the particle is trying to get inside the cell, and it kind of burrows up in the side, and a vesicle is formed. And when that vesicle is formed, it is released inside the cell. 3 types of endocytosis. We have phagocytosis, which is cellular eating. Solids are engulfed, such as your bacteria. We have penocytosis, that's cellular drinking. Liquids are engulfed, such as your lipids. And then receptor-mediated endocytosis. This takes up specific molecules targeted by the receptors. The cell must recognize it before it can enter. Then we get into cell division. So cell division, we're going to talk about mitosis primarily, but I will mention to you what meiosis is. But with mitosis, we have cell division that results in 2 identical daughter cells. So 46 chromosomes. This enables growth and repair of our tissues, and it does occur in somatic cells. Now with meiosis, this is cell division that results in the production of ova and sperm. 4 daughter cells are produced that have 23 chromosomes, and this occurs during our reproductive cells. So we're going to focus primarily here on mitosis. Now mitosis forms diploid cells, while meiosis forms haploid cells. And remember, mitosis is the division of the nucleus, and again, it forms after cytokinesis. So with mitosis, the mitotic cell division produces 2 daughter cells with identical chromosomal content as the parent cell. So here are the 3 stages of the mitosis cell cycle in somatic cells. Now we have interphase. Notice here, interphase is DNA is in the form of chromatin. So interphase is simply, one, and you can see here in the circle in the drawing here, in the center, we go all the way through our growth, our DNA synthesis, growth again, and then we get to mitosis. Now, mitosis itself, these steps come in this order. It's prophase, metaphase, anaphase, telophase. Then when we get through with mitosis, we get to cytokinesis. So with mitosis, that's where we have the division of a cell's nucleus. Chromatin is in the form of chromosomes here. And again, we are trying to replicate the DNA, the chromosomal material inside that nucleus, and make 2 identical daughter cells. So when that is finished, we go through cytokinesis, which is the cytoplasm of that identical cell pinches in half, and that's where the cell splits, and we have 2 identical daughter cells with the identical genetic information as a result. So make sure you know it in that order: interphase, then prophase, metaphase, anaphase, telophase, then cytokinesis. So interphase is the first stage of the cell cycle. It is a period of cell growth and development. The cell is performing its normal functions and preparing to divide. So here's where we have our DNA replication. So our DNA is being copied inside of the nucleus. Now the second stage of the cell cycle is, of course, mitosis as a whole, but in that process of mitosis you have prophase, and that's where the chromosomes will form. The nuclear envelope is going to start to dissolve because we need that DNA information inside that nucleus to start to separate, and it's already been copied, it's already been replicated. We need it to separate into two different copies. So the centrioles are going to migrate to opposite poles, and the spindle fibers are going to develop. Now, spindle fibers are specialized microtubules that guide the separation of chromosomes, and they are formed from the centrioles. Now, after prophase occurs, we go through metaphase, and all the chromosomes have separated, and then all of a sudden in metaphase, they start to line up in the middle of the cell. Now, remember, our whole goal is that we've started with one cell, we've replicated and copied our DNA in interphase, and then we're trying to split it into two identical copies and then go through cytokinesis where the actual cytoplasm splits and we have two actual cells as a result of one. Anaphase is where the sister chromatids separate to opposite sides of the cell. And then telophase is where the chromosomes reach opposite ends of the cell and new nuclei start to form. And you'll see the cleavage furrow in telophase, and that's the indentation at the equator of the cell. So here's a picture of prophase. Again, all the chromosomes have already been replicated in interphase, and they're starting to separate here in prophase. And the picture on the right-hand side is just a microscopic view. And then, of course, the left is an animated drawing. But here in metaphase is where we line up along the center, along the equator of the cell. And again, notice here on your chromosomes to the left, you see two actual identical copies of each. And you can also notice your spindle fiber is formed, and notice your centrioles. Remember, centrioles were only there in time of division. And then anaphase, notice your two copies are starting to be pulled apart. They're starting to separate, and again, our goal is to make two identical cells, so they're starting to separate to opposite sides of the cell. And then notice in telophase, the chromosomes actually reach opposite ends, and nuclear envelope starts to reform around them. Notice the cleavage furrow, which is the indentation of the two cells joining. That is called a cleavage furrow. And what's going to happen next is cytokinesis, where they actually pinch in half. Now, this picture to the right is still telophase. That's not cytokinesis, but cytokinesis would look like two complete, like they would not be attached at all, cells right next to each other. So the end result, of course, is two cells with the same genetic information where only one existed before. And these are just some microscopic views that we would see if we were in the lab looking at the microscope. But you can see here tons of different, we've got interphase going on. We see some prophase. I see an anaphase. I see a metaphase. So lots of different activities going on there with all those different cells. Same way here at the very, very top, I see that is actually telophase, where you have the cleavage furrow, and you can see the indentation of those two cells that have formed, and they have not split yet, but when they split, of course, that would be cytokinesis. So that is chapter 3, the cellular chapter.